this is about how scientists think. Scientists think in a way which is called the scientific method. Common people like to make assumptions and they like to make conclusion without any solid data. But scientists perform experiments, analyze their data, and then make conclusions. Now let's look at what are the steps of scientific method. First is observation and asking question. Let's say your phone stopped to work. The question would be, why is my phone not working? In a more scientific scenario, the question would be, are steroid medications effective in relieving asthma? You will look at the past studies or past experiences. Has anybody worked on similar situations or has faced similar situations in the past? So, has anyone experienced this situation before? Was there an effect that was recorded, maybe from your family or friends? You hear that a phone stops to work when there is water inside your phone or your battery dies. In a scientific scenario, you would like to know, has anyone tried to use steroid medication in relieving asthma? Based on your background knowledge or whatever little information you have about this situation, you state hypotheses. Hypotheses is a calculated guess or an educated guess. There are two kinds of hypotheses. In the scientific term, first is null hypothesis. A null hypothesis is a no situation hypothesis in which you say there is no difference in the factors, there is no effect of the factors, and there is no correlation between what you're seeing and the previous information that you have. So a null hypothesis for your phone situation would be neither the battery nor water has an effect on your phone dying. An alternate hypothesis is where you consider some factors and you're going to test whether those factors are the reason for your situation or not. For example, you would say alternate hypothesis is the battery is making your phone to die, or water is the factor that is leading to your phone not working. Then you collect the data, you perform experiments and see which factor is responsible for the situation. For example, you would change the battery, you would remove the water from your phone, you would see if there is any software update needed, you would change the cable of your phone and see if there is some overheating situation. In a scientific situation, you would perform all the experiments, you would do all the tests and see what is the result that you're seeing after changing these factors. So analyzing data is actually looking at the results, analyzing which of these list of factors is responsible for the change. It might be one of the things that on changing which your phone starts to work. And in a biological or a scientific scenario, you would look whether your patient has gotten better after giving medication, gotten worse, or remained unaffected. Then, after performing the experiment, after analyzing your data, you state a conclusion. You would pick one of the scenarios, whether it was the phone battery or water getting into the phone, or none of these factors was responsible for the observation that you had. If you're not able to make any conclusive results, then you go on and develop more hypotheses, try to guess more factors that could be leading to this problem, because your goal is to solve the problem, solve the situation. Because you want your phone to work, then you would keep on testing other things that are responsible for your phone not working, or the scientific question that you have, you want answer to those questions, so you would keep testing your hypotheses. So in brief, hypothesis is the guesswork you do based on the previous information before doing your own experiments, and then you do your own experiment, and conclusion is what comes after the experimentation and analysis of data. So to summarize, these are the 7 steps of the scientific method, which is observation and asking question, performing background research, stating hypotheses, doing experiments, collecting data, then analyzing data, then stating your conclusions. And if you do not have any conclusive results, you keep on performing your experiments based on your newer hypotheses. Let me give you an example of one of my studies where we had used some background information to develop a hypothesis to study the mode of action of a drug. So let me give you the background of my research first. This is the parasite that I was working on. This parasite is called Trypanosoma brucei. Just like a malaria parasite, it is transmitted by the bite of an insect. It is more prevalent in sub-Saharan Africa. In human beings, it causes sleeping sickness, and in animals, it causes this wasting disease in which the muscles get wasted. So we had found a drug, let's call that drug Sharma drug. We had infected mice, and then we gave some mice no drug and some mice my drug, and we wanted to see whether the parasite has reduced or not. So we had some genetically engineered parasites which glow just like firefly. So the color that you're seeing is representing the parasite glowing just like firefly, and more the color, more the parasite. First 5 animals, V1 to V4, were not given any drug, and N1 to N4 were given the Sharma drug. As you can see, the animals given Sharma drug have lesser color, lesser signal. That means they had lesser parasites after treatment. So we knew that the treatment was effective. Now, how does this drug kill the parasite? That was my question. What background knowledge did I already have? So we had already performed a proteomics experiment in which we knew that this Sharma drug destroys a protein which is required for the synthesis of DNA in the parasite. Because we already know that there was a hypothesis generating proteomics study already done in the past. The question or the hypothesis then became, either the Sharma drug does not affect DNA synthesis, or in alternate scenario, the Sharma drug would either increase the DNA synthesis or decrease the DNA synthesis. Then we had to perform the experiments and analyze data. So here is the parasite that we were working on. The first group of parasites were given no drug. The second set of parasites were given Sharma drug, and third sample of parasites were given another drug which does not have a similar mechanism of destroying DNA synthesizing proteins, but a different mode of action. So what you're seeing in second panel are the nuclei of these parasites. So this is the nucleus of these parasites. I'm sorry, this is this one, and this is this one. Similarly, this is this guy. The nucleus of all these parasites is represented in blue. We had a technique in which the DNA could be tracked in green color. So out of the 4 parasites, 3 were synthesizing new DNA. But after treatment with the drug, 3 parasites, out of the 3 parasites, only 1 was actively synthesizing DNA. Again, in the old drug, which does not affect DNA synthesis, 3 out of 4 parasites were synthesizing DNA. That means we had to analyze this data, and we saw that in the group that was not given any drug and in the group that was given a different drug, there was almost equal amount of DNA synthesis. But in the drug that Sharma or I was using, we saw a reduction in DNA synthesized. Let's go back to the hypotheses that we had. So we had three hypotheses. The null hypothesis, which said there is no effect on DNA synthesis. There was hypothesis 1, which was the Sharma drug would lead to reduction in DNA synthesis, and there was hypothesis 2, which said that the Sharma drug would increase the DNA synthesis. After experimentation and analysis of data, we found that Sharma drug actually reduces the DNA synthesis. So our H1 hypothesis was the one that stood true. And these findings were then reported in a scientific journal. But if we had not found any conclusive results, we would have kept doing these experiments and kept designing more hypotheses. So this is how scientists think about a problem, do experiments, analyze their data, state a conclusion, and then those conclusions are reported in scientific reports.